Welcome everybody to today's session on From Species to Ecosystems, Integrated Population and Speciation Genomics. I'm Richard Durbin and I'm co-chairing this session with Nina Therkildsen. So we have 4 pre-recorded talks and then there will be a panel discussion and a question session. And the 4 talks, the 4 speakers will be Priscilla Saloom, Andrea Guarancino, Nina Therkildsen, and Michael Westbury.
And during the talks or after, please go ahead and add your questions to the Slido chat box below. And since we only have 4 talks in this session here, we'll have a bit more time for discussion at the end. So please go ahead and add your questions. Then also, we just want to make sure you're all aware that that's, in addition to the talks, there's a set of really great posters associated with this symposium. So we strongly encourage you to check those out at a later time. So let's jump right into the talks.
Kia ora, hello everyone. My name is Priscilla. I'm a research fellow at the University of Auckland, and today I'm presenting my current work on assembling the genome of a marine mollusk. We live in an era where genome resources are increasingly accessible even for non-model species. The use of varied genomic methodologies and large datasets have improved our power to infer the distribution of neutral and adaptive variation and the evolutionary forces underlying this variation. Significant contribution to this knowledge has come from the possibility to generate a large number of SNPs without the need to have a complete genome assembly as a reference, and using approaches such as genome scans to disentangle background and non-neutral differentiation in specific regions of the genome. This is what we did for the New Zealand Urchid chiton, Onychochiton neglectus. Using genotyping by sequencing, we generated SNPs genome-wide for 16 populations of Oneglectus, comprising most of its distribution range, from the warm northern New Zealand to the subantarctic islands. As you can see, this approach enabled us to identify strong subdivision into 3 clades: Northern North Island, Central New Zealand, and a southern clade, including populations from the south and the subantarctic islands. Such strong population structure between clades can be justified by life history traits. Oneglectus has very limited mobility as an adult, and it lacks a long-lasting pelagic larval phase. Females brood eggs until they hatch, and settlement happens nearby within a few hours from hatching. This might very well explain the low genetic connectivity in the north and central clades, but not why populations so far apart between the South Island of New Zealand and the subantarctic islands show stronger genetic connectivity and are part of the same clade. The key information here is that in its southern end, Oneglectus can hitchhike in the holdfast of floating Dovidia kelp for long distances, and this mechanism enables some connectivity between populations so far apart. This phenomenon is rare in lower latitudes as populations of Dovidia kelp are scarce up north, and there are no reports of Oneglectus associating with other seaweed species. In this complex population structure scenario, it is tricky to apply genome scans, but with careful statistical considerations, we were able to detect interesting patterns in allele frequencies among the 3 clades, and with some alleles differentially fixed in populations of each clade. Also, some loci within the southern clade are likely to be evolving non-neutrally, showing correlation with the latitudinal variable marine environment of New Zealand and indicating local adaptation. The GPS-generated SNPs were very useful for identifying the patterns I just showed you. However, a reference genome would be very beneficial to enable further assessment of these patterns. Mapping the population-wide SNPs to the genome will allow determining polymorphic versus conserved regions across the 3 clades that could be under diverging or constraining selection, as well as checking if candidate loci previously identified fall within functional genomic regions, and from there predict the selective factors that could be acting on them. So we started to assemble the genome of Oneglectus, and the idea is in the title of this talk, a high quality and low budget genome. The only other chitin genome assembled is relatively small, so we predicted that this would also be the case for O. neglectus. To achieve a high quality and low budget genome, we were going to use 2 flow cells in a MinIO-ONT device and combine these long-range reads with short reads coming from Illumina sequences. We expected that we would get around 60x coverage with the long reads and 40x coverage with the short reads from 26 giga of data. Finally, I have submitted RNA for sequencing, and I intend to assemble transcriptome de novo and try to use it to help with annotation. As with all the projects, we faced a few unexpected challenges on the way, particularly in the wet lab. Extracting high molecular weight DNA is one essential first step in genome assemblies. And when we thought we had finally achieved that, we realized that we were actually co-purifying contaminants, and no amount of cleaning would remove that. We reached out to the amazing community working on chitins and other trouble organisms and learned that it is common to co-purify such contaminants in the sticky mollusks and other sticky marine critters. Continuing to optimize our protocols, we added beta-mercaptoethanol to the lysis buffer of a previous protocol, the NEB high molecular weight extraction kit, and also tries the CTAB buffer, CTAB protocol with and without a pre-wash using sorbitol. We have got a few libraries that seemed contaminant-free, and then a very happy day in the lab when sequencing worked. But we noticed a compromise on read size, mainly for the libraries coming from the CTAB protocol. So our current strategy includes using short read eliminator kit for with a cutoff of 8 kilobases, and then sequencing a library produced by the CTAB protocol because it gives a good depth of sequencing, and then flush the flow cell and sequence a library generated with the NEB protocol, as this library has longer DNA fragments and it helps increase the N50. And we have got better results using this protocol than our first attempts. The assembly is slightly larger in size than we expected, and the N50 is much better than the 19 kilobases of the first trials. These stats are okay for the amount of data coming from one flow cell, which is less than we initially expected. This is a work in progress, and we will continue to follow a strategy and increase our sequencing efforts. As an exploratory step, I used RegTag to align the current O. neglectus assembly to the Acanthopleura granulata genome, a chitin in the same family as O. neglectus, but with a number of different life history traits. RegTag aligned 43% of the O. neglectus bases to A granulata genome and improved the N50 of the O. neglectus assembly to 230 kilobases. The A granulata genome is very contiguous and has high quality, so it helps my fragmented assembly of O. neglectus at the cost of inserting many more missing bases throughout. We will use another MinION flow cell to increase the amount of long reads generated, which will not increase the cost of the assembly that much. And I'm also about to send DNA for Illumina sequencing, but was held back by the COVID lockdown here in Auckland. Once I have a better assembly, I can move on with annotation and mapping the population data to the final assembly. And now I would like to thank you for listening, and also thank my co-authors and collaborators, Annabelle Webley, Anna Sancho, Shane Lavery, and Pierre de Villemihir, and also acknowledge the computing resources provided to this project by the New Zealand Youth Science Infrastructure and funding by the Malacological Society of Australasia and the Kate Edgar Educational Charitable Trust. Thank you.
Hi everyone, I'm Andrea Guarraciro, a PhD student at the University of Rome Tor Vergata. Today, I will present our preliminary results in studying chromosomes communities in the human pangenome. Thanks to advances in sequencing technologies, high-quality de novo genome assemblies will become very common in the near future, and the huge amount of data will offer the opportunity to study genomic variation as never before. However, lots of data also bring challenges in how to work with hundreds of genomes at a gigabase scale. A pangenome can model the entire set of genomic elements in a population. Pangenomics then contrasts with reference-based genomic approaches, which relate sequences to a single linear consensus model of the genome. In particular, genomic approaches are biased because the genomes reconstructed with the help of a reference genome appear more similar to the reference than they actually are. Pangenomic approaches can reduce this bias by relating each new genome to all the others represented in the pangenome. In particular, genomes can present regions that are not inalienable against the reference, and so cannot be represented in a list of variants. Graph-based data structures overcome this problem by capturing in a very compact way the sequences and their relationships. In pangenome graph models, DNA sequences are incorporated as nodes, with edges connecting the nodes as they occur in the sequences represented in the graph. If some sequences present identical regions, such as paralogs or orthologs, they will share the same nodes, while the variants will be represented as new branches in the graph. To date, among the high-quality de novo assemblies available for the human species, there is the data of the Human Pangenome Graph Consortium. This consortium aims to issue a new pangenome reference that reflects the full range of genomic diversity across the human population, avoiding the ethnic bias present in the human reference genome. The HPRC dataset consists of telomere to telomere phase deployed de novo assemblies from 47 individuals. In our research, the contigs are partitioned by chromosome by mapping the assembly against the two human references. These references are also embedded in the pangenome graphs we construct for using them as coordinate systems for liftover operations. This data is under extensive quality controls. One of the analyses performed by Hank Lee involved the identification of disjoints by mapping the contigs against the CHM13 reference. The slide reports a visualization of its results as an alignment graph, where nodes represent the contigs and the edges represent how many mappings between the contigs are obtained. Except for one sample, all the misjoins involved the acrocentric chromosomes, suggesting the presence of homology relationships between non-homolog chromosomes. Therefore, we decided to go further in this analysis, but in a reference-free manner, by aligning all contigs against all the others. We then constructed an alignment graph by considering only the mappings longer than 1 megabases to reduce possible mismappings. Finally, we applied a parameter-free community detection algorithm to explain the mapping network as a set of communities. In particular, we were interested in groups in which the two human reference sequences are together. On the right, there are the results we obtained. We highlighted the two acrocentric communities and also the group with the chromosome X and Y. Sex chromosomes are expected to appear together because of their pseudo-autosomal regions where they recombine. These results are stable across different bootstrapping experiments based on sampling several thousand alignments and recomputing the communities. On the left square, there is a visualization of the alignment graphs made with more stringent mappings to obtain a graph enable to visualize. Chromosomes are relatively isolated with a handful of connections to others, except for the acrocentric communities, where chromosomes 13 and 21 result to get closer to each other as chromosomes 14 and 22. When we construct pangenome graphs for these chromosomes separately, this is what we obtain. The layout shows the graph topology with the regions of variations that appear as bubbles. The complex motifs on the left are due to the difficulties to assemble and align these highly repetitive sequences present in the P arms of these chromosomes. However, if we process all the contigs that belong to the acrocentric chromosomes together, this is the community pangenome graph we obtain. The layout shows a large connected component with the P arm sharing a region of high similarity. The CHM13 reference genome confirms this high degree of similarity between the acrocentric chromosomes, as shown by the structure of its assembly graph on the left. These results matches what we observed in the alignment graphs. Indeed, if we take a closer look, we can notice two bifurcations highlighted with black circles in the graph layout on the right. These bifurcations are due to the high similarity that chromosome 13 and 21, and also chromosome 14 and 22, share across the centromere and pericentromere. The image on the left shows the p-arms of these chromosomes. For each chromosome, the percent identity heat map versus the other 4 p-arms are reported below the annotations. The red heat maps indicated by the colored arrows show the high similarity regions that lead to the bifurcation in the pangenome graph. All these evidence goes in the direction of the putative presence of an exchange of sequences between non-homologous chromosomes, possibly due to an evolutionary process that had already been hypothesized several decades ago. In our research, we still have to find strong direct evidence of this type of recombination in the human population. P-arms of the acrocentric chromosome are difficult to study because of their highly repetitive sequences that complicate assembly and downstream analysis. However, high-quality de novo assemblies and new pangenomic approaches will open the door to also these regions, allowing us to decrypt possible traces of recombination between non-homologous chromosomes. Thank you for your attention.
Hi, I'll be talking about how low coverage whole genome sequencing is now emerging as an increasingly powerful and actually surprisingly cost-effective approach that I think has really versatile applications for population genomics. So we all know how the cost of DNA sequencing has dropped dramatically over the past few decades. But for any project, researchers are still faced with the inevitable trade-off of having to decide how to divide a given sequencing effort between the breadth of genome coverage, how much of the genome are we going to sequence, how deeply are we going to sequence each part of the genome, and how many individuals do we want to sequence at that depth. So especially for non-model organisms, a typical approach has been to prioritize deep sequencing in a large number of individuals for a small random selected part of the genome. And that's a very powerful approach for, in a cheap way, getting high quality genotype data distributed genome-wide. But with the relatively sparse sampling of the genome, this approach, such as, for example, RAD sequencing, can leave us wondering what is going on, what might we be missing in the parts of the genome that we're not sampling that are located between our markers. To get at that, we might instead choose a different approach of spreading our sequencing across the entire genome. But if we want to still analyze the same number of individuals, that comes at the expense of sequencing depth. So when we do really low sequencing depth, we are not able to reliably call individual genotypes. But fortunately, over the past decade or so, there's been a really great advance in the toolbox that we can use to still make robust inference in downstream analysis, as long as we conduct it in a probabilistic framework that takes uncertainty into account. So I want to quickly show you a few examples of how we've been using this approach. The first being a study of adaptive divergence in Atlantic codfish in the Gulf of Maine. And this is work that we've done in collaboration with Adrian Kovac's lab at the University of New Hampshire. So we sampled more than 300 fish from 20 different locations. So small sample sizes, less than 25 per location, and all sequenced to an average of just under 1x sequencing coverage. And here we're looking at a series of pairwise comparisons between populations. On the y-axis is FST, a measure of genetic differentiation, and it's shown in genomic windows along the genome on the x-axis. So the first thing we notice is that genome-wide, there's virtually no differentiation between many of these populations, but we see these huge blocks that are known chromosomal inversions in cod that are showing highly elevated differentiation. And these patterns, even despite our very low sequencing coverage and low sample sizes, are showing up really clearly here. But what we're also seeing is this narrow region of differentiation of divergence that's highly localized in the genome. And this particular peak here actually differentiates populations that spawn in the spring and the winter. So it's a biologically meaningful signal. But it's so narrow that with sparse sampling, for example, RAD sequencing, we might completely have missed this. As a case in point, there actually was a RAD sequencing study comparing some of these populations earlier on. And we see they did happen to find a single SNP marker in this particular peak. So that's something, but with just a single marker, it's hard to make any robust inference about whether that's a real signal or not. In contrast, even with our whole genome low coverage data, even though we don't have any reliable individual genotypes, we have much more resolution to infer that this is a real biological signal. Another fish that we've been using the low coverage sequencing approach to study is the Atlantic silverside. And this is work I've done in collaboration with a number of postdocs and students in my lab, and with Hannes Baumann, Steve Palombi, and David Conover. So silversides are distributed across one of the world's steepest thermal gradients on the east coast of North America. And it shows a remarkable degree of adaptation to local conditions. And similar to what I was showing in the card, by using low coverage whole genome sequencing, we've discovered that even though there's very little genome across distribution range, there are certain big chunks of the genome that we now know to also be chromosomal inversions in this species here that show extreme levels of just extremely high differentiation. So this kind of inference we could also have made with pooled sequencing, which is when we just mix the DNA from multiple individuals in an unlabeled way to a single library. But the approach that we've been using of sequencing individually barcoded DNA from different individuals with low coverage of these, we are able to recover some really valuable individual information that would be completely lost with a pooled sequencing approach. So for example, here with data, low coverage sequence data from individuals from 11 populations, we are able to get very clear signals in a PCA. And what we can notice is that some individuals fall in this PCA space very far from the other individuals from their populations, suggesting to us that these may be long distance migrants. Some inference that we'd never be able to make if we had pooled the DNA into a single library. Also, even though we're not able to get individual genotypes reliably, we are still able to, within these big inversions that I showed before, show highly elevated differentiation. We can still see, if we make our best guess at the genotype at each of those SNPs in individuals from different locations, we can still recover a really clear signal of strong linkage disequilibrium in these blocks and a really clear haplotype structure that we can also then compare the frequencies of different haplotypes in different populations sampled up and down the coast here. In a different study where we were looking at the genomic basis of response to size selection, we were also able to look at correlations between individual genotypes across large sets of SNPs that we expected to have a polygenic effect. So even though, again, no reliable genotypes at individual SNPs, when integrating over the uncertainties in larger SNP sets, we could look at genotype-phenotype relationships. We could infer individual levels of relatedness. And importantly also, when we sequence to low coverage at the nuclear genome, we actually get really high coverage of the mitochondrial genome because of the much greater copy number in the cell. So this means that we've also been able to recover high confidence whole mitochondrial genome sequences for all these fish that lets us conduct very powerful phylogeographic analysis. So I think there are a lot of advantages to this data type, but how hard is it to do? So there are really well-established methods to do a simple and quick library preparation. The biggest challenge is that we need to adopt a different toolbox where we need to do all our downstream analysis based on genotype likelihoods instead of actually called genotypes. So we take the uncertainty into account. But there's a growing toolbox available that can handle these types of data. And just a little shameless plug here for anyone wondering how to get started with these. We recently put together a guide to getting started with low coverage whole genome sequencing, where we review available software for different types of population genomic inference. We also did a series of evaluations on simulated data to look at the trade-off between the sequencing depth per individual and number of individuals, how we could get the greatest inference power to estimate allele frequencies, population structure, and FST outliers. And overall, our conclusion was that in general, spreading the sequencing across more individuals with lower depth per sample improves our inference accuracy much more than that having higher sequencing depth for fewer individuals, except from a few types of inference that are notable. And in terms of cost, at this point, sequencing is so low that for many species with typical genome sizes of mammals, birds, we can do this type of analysis, including the sequencing, for less than 40 or 50, and typically much less than that, even $15, $20 per sample, which is really comparable cost to rat sequencing that gives us a much lower genome sampling. So I think we're at a point now where low coverage whole genome sequencing has really matured to become a very viable approach for population genomic studies, and that it allows us full genome resolution while retaining many level inference, and at a similar cost to both pooled sequencing and rat sequencing. So I think it's very exciting, all the insights that this approach is going to provide in the future. With that, I'd just like to acknowledge collaborators and thank you for listening.
Today, I'm going to talk about our recent work that we have done that looks into the role that the reference genome, and specifically the phylogenetic distance of the reference genome, can have on evolutionary inferences. So everything that I present today has recently been published in Molecule Resources, and here is the title, so anyone can look it up and get a lot more information if they wish. So a brief introduction into the different types of assemblies, which I'm sure many are aware of, but so we have mapping assemblies, where you just map the short reads of your individual to a already published or already assembled genome. And this method is relatively quick and easy and inexpensive compared to the alternatives, at least. And the downfall, though, is that it requires, a published genome or a genome of a con-specific that you've already assembled. But the question is, do we really need the con-specific genome? And we also have de novo assemblies, which is essentially making a new assembly, a new genome from scratch from raw reads. This method is a lot more time and computationally expensive, but it's also very monetary expensive compared to mapping assemblies. But the benefit of this is that you get the exact species of interest, get a genome from the exact species of interest. However, there's also one alternative method that has recently becoming more popular, I guess, or more available, is cross-species scaffolding, where you create the contig level assembly from your species of interest. So just using the short Illumina read data to make a rough contig level assembly, and then you use the distance information from a related species with an assembled high quality genome to scaffold the contigs. So essentially put the contigs together and put ends or missing data between the contigs, so you have more contiguous assembly, but not really adds new nucleotide information. This is also relatively time and computationally consuming, but the benefit is it's inexpensive. And the method that I used, or that we will talk about today, is the method by Grau et al. And here's the paper for those interested. So the aims of the study was to look at how the reference genome, the selection process, can influence either demographic history, heterozygosity, or inbreeding estimates in the form of runs of homozygosity. And we looked into increasingly distant reference assemblies, and by mapping to increasingly distant reference assemblies, but also mapping to newly assembled cross-species scaffold assemblies that we assemble for the study. And for the study, we used two different study systems. One was a mammalian one, which was the beluga whale and a lot of other cetacean relatives. And one was an avian one, which was the roe kiwi with other kiwi relatives and also the emu. So the phylogenetic distance of our target species to the other reference genomes that we had ranged for the beluga from about 0.5% all the way to 3.4%. And for the roe kiwi, it ranged from its closest relative, the brown kiwi, of about 0.3% all the way up to about 7% with the emu. So this is absolute genome divergence, if you will, pairwise distance. So the way we conducted the study was first we obtained the reads from our target species, so either the beluga or the roe kiwi, and mapped that to the published assemblies I just showed there, and conducted the analyses based on that. Or we took those raw reads, created a contig level assembly, and then used cross-species scaffolding to put the contig level assembly together in different ways, one for each different reference genome. And so we ended up with essentially a beluga or a roe kiwi assembly, but the distance information was slightly different. And then we mapped to these cross-species scaffold assemblies and conducted the same sort of analyses and compared that to mapping just to the published assemblies. So the published assemblies were all relatively high quality, ranging from, for the cetacean species, about 6 MB all the way up to chromosome level 122 MB N50s. The kiwi and emu were not quite as high quality, but still relatively high quality of around 1 MB, at least 1 MB up to around 5.7 MB. The cross-species scaffolded assemblies, on the other hand, were very fragmented relative to the published assemblies, especially for the cetacean, where we're around 300 kb up to 600 with the closest relative, and then a lot of missing data was also included. And the kiwi was also very similar to the published assemblies. So for the demographic inferences, you could see that the black is the species of interest, and this the names here are listed in order of phylogenetic distance. And as you get more and more phylogenetic distance, you see a deviation from the expected pattern, but you still see in the overall kind of flow of the graph. And very similar with the rowie, but not quite as pronounced until mapping to the emu. On the other hand, when using the cross-species scaffolded as reference, you can see that the pattern is also kind of the same, but all of the mapping assemblies are deviated equally from the original result. But this is not really the case with the kiwi, so that seems to be more reliable. And this may be just due to the differences in assembly quality that I mentioned. The cetaceans were a lot lower quality. So with the heterozygosity, we see also an increasing deviation from the expected here as phylogenetic distance increases, but not so much with the kiwi until you hit the emu, which is a lot more distant. When using the cross-species scaffold assemblies as references, that deviation essentially completely disappears, slightly still there for the most divergent, and it also relatively disappears for the kiwi data set. What I didn't show here is that we also tested a bunch of different parameters and software, and the results still showed these biases. So it wasn't just the software or the parameters that I selected or just showed. We couldn't look into runs of homozygosity in the beluga because there were none, but in the kiwi, we found that with the published assembly, we were getting runs of homozygosity larger than 1 MB, about 4% of the genome. But when we mapped to the non-conspecific, we were essentially just losing those, but we managed to recover those with cross-species scaffolding. So in conclusion, the demographic history and heterozygosity were deviating, correlating with phylogenetic distance of the reference. And also the runs of homozygosity essentially just disappeared when mapping to a non-conspecific reference. However, we also showed that cross-species scaffolding, even though there are lower assembly qualities, could be a useful substitute when nothing else is available. So thank you for your attention. And here is the paper again for all of those who might be interested.
Yes. Thank you very much, all the speakers. There are a few questions beginning to appear. Please keep on adding questions. There's several for Nina. So Priscilla asks whether it's possible or difficult to look for structural variants when you have low coverage sequence.
Well, so definitely, so as I was showing in my talk, to big inversions like what we see in both cod and silversides pop out, but mostly they pop out because they show elevated differentiation. So one thing that we're kind of curious about is how many of similar size to other big inversions are there in the genome that are not showing elevated differentiation that we might be missing just because we haven't looked yet. So it is kind of limited. We definitely have much less power to detect structural variants than we would have with high depth data. But so I don't have a good answer because it's something we're still kind of exploring, but at least when any structural variant that's reasonably frequent in a population, my intuition is that we should still have, even if the reads that we have are from lots of different individuals, as long as we have a per population depth of 30 or 50x, there's information to work with, but it's definitely a lot more challenging having depth data for individuals.
Yeah, and I think, having explored this a bit myself, I think it's really a question of, so if you're looking for copy number deletions or duplications, there's a sort of trade-off of size. So you can still see long copy number variants with quite high accuracy with low coverage sequence. And in fact, Illumina data is not good for some sorts of structural variant anyway. So yeah, it's degraded, but it's not bad. Okay.
Okay, then maybe I'll ask Richard's question to Andrea. Is it right to say that the Q arms of the acrocentrics form well-separated standard chromosomes, while the P arms should be pooled together conceptually? And are the relationships between the P arms different between different individuals?
Yes, it is correct that the P arms are pretty similar. In the last CHM30 reference sequence.
You're sounding a bit distant, Andrea. I wonder if your microphone's working.
Now better?
Yeah, better.
Okay, so I was saying that, yes, it is correct to assume that. Indeed, in the last CHM30 reference sequence, and confirmed this similarity, and also in the Pancino graphs, in the human Pancino graph that we are building, always the Q arms remain separated. We also try to make Pancino graphs with the all chromosomes, human chromosomes, and they stay separated. And we always get a single connected component where the acrocentric chromosomes collapse in their P arms. About the population, that's the point here. We don't know what it is happening in humans. So we still need to identify variations in the P arms. The problem, as I said in the presentation, is that in that regions, the assembly could be wrong, could be enriched in misassemblies. And also the tools that we are using and developing, like the mappers, for example, the repetitive sequence structure makes this task really, really difficult. So that's the current issues to tackle.
I'm wondering if you could use Hi-C data to link variants in the P arm to variants in the Q arm to address this. I think that HPRC has Hi-C data for all its samples.
what do we want to do? The next steps are we need to find traces of recombination. A way could be if we would be able to call enough, with enough confidence, the variants and find contexts that span the entire centromeres. We would like to see, okay, I have this pair of variants. I see this variant in few individuals and they stay together, always together. Or we found a recombination. Okay, I have two variants in different chromosomes, in different arms. And yes, we are going to use also that type of data. But first, we would like to try to use the current context. That's the current plan.
Okay, so I have a question from Sibel Vilasa to Michael. She says, thanks for your talk. Do you have an idea why heterozygosity estimates are higher with distance to the reference genome?
Well, it's a really tricky one because it kind of goes against the opposite of what is currently thought. So I don't really have a really great answer for that. Maybe Richard actually could have something. But I'm thinking it's something to do with just more misalignments with the mapping tool itself, not being able to align quite as correctly to the more distant references.
I'm wondering if it's heterozygous, maybe heterozygosity increases. Yeah, don't know. I was going to produce an argument for the reverse, but
well, if we use
have the empirical data, so
if we use the back parameter, it actually decreases with distance. So it's really kind of parameter specific as well. So that corrects for like multiple misalignments in the reads to the reference.
Mm-hmm.
So is it because private alleles in the other species you get more private alleles with greater distance, and then they become, if they're heterozygous in the individual? Or
yeah, I mean, no, it must be either that you're losing homozygous regions, or that would increase the apparent heterozygosity, or you are synthetically mapping two things which really are the same, together to make fake false positive hets. I don't know. But I think the message is kind of clear that you can create artifactual distortions by mapping to a distant reference. I note that in a lot of invertebrates, those distances that you're talking about of a few percent, which you will also occur within species, so that that it's quite common to get several percent heterozygosity. In fact, maybe I should ask Priscilla what the heterozygosity is in your chitin. I think that could be several percent.
Yeah, actually, it's not very high at all. When looking at the SNPs, I estimated observed heterozygosity, and it's usually below 0.1, sorry. And when I look at the individual groups themselves, it's about 0.2. So I don't expect the genome to be very high either, but I didn't get to that point of the analysis yet.
Okay. I was seeing 2 to 3% heterozygosity in some Darwin tree of life chitins, but which we haven't yet assembled, but
yeah, I don't know if the
different species probably different.
Yeah, the breeding behavior makes them so not able to disperse that much, and they end up, I don't know, inbred perhaps, but yeah.
Okay.
I was curious, Priscilla, those contaminants that you've been struggling with, do you think, are those found across lots of marine invertebrates, or are many of them more restricted?
So I heard a lot of people complaining about contaminants and not being able to get nanopore working on marine critters. I think mostly the some mollusks and potentially worms as well. And people just come up with the most creative ways of removing them sometimes, or they just change the platform for sequencing. And yeah, I think it depends on what you were working on. And sometimes you plan for something, and when you get to the lab, you realize it's actually a bit more work than you thought before. So yeah, I think for us, the big constraint is the low budget part of the project. So I think you just have to give it a try and talk to people before you even start your project when you are working with a true organism like that. But yeah, those are usually polyphenolics and mucopolysaccharides and stuff like that.
Okay, well, Nina, there's a couple of questions for you. So one is from Fernanda Trindade, and she asks, what happens if you try and use the usual toolbox for SNP calling on low depth data? Are there cases where it can be reliable?
I think in general, I'd very strongly recommend against it. Just when we're sequencing as low as 1x or lower on average, then even for the average SNP, we won't even be able to detect a heterozygote. So inevitably all genotype calls, it is going to be super noisy. And then I've seen some people then filter to only use the sites that have higher coverage, but then obviously you're biasing to looking at certain parts of the genome that sequence more efficiently, maybe because they have different GC content or whatever. So it seems to me that now actually the toolbox for incorporating genotype uncertainty into downstream analyses has been so well developed that there are most of things, most of the types of analyses that you want to do. Then I think if there's some ways maybe you can do some downstream processing on, for example, take allele frequencies and run with them for different downstream analyses, but I don't think there are any great cases where you get reliable genotype calls from very low coverage data. I don't know if anyone else want to weigh in on that.
No, I can make a comment that, so I also work in ancient DNA a bit, and of course there you have lots of very low coverage samples. Mm-hmm. And they do a lot of population genetic analyses and using a variety of approaches. So some are using likelihoods with angst and so on, and some are using these pseudo-haploid measures where you take a random allele variant read wherever there's an overlapping read, which is a sort of random sample essentially. So yeah, I support that there's lots of machinery out there. Are you using angst? Is that the toolkit that you're talking about?
Mostly angst, but we tried in this paper I mentioned we just put out, we tried to also highlight lots of different other sort of standalone programs that people are developing for specific purposes. Mm-hmm. And I think in many cases, a lot of other software can also be rewritten to take genotype likelihoods as input instead of the actual genotypes. So I think there's more and more interest in adapting tools to be able to use this data type, and also becoming more and more of a user base.
And then the other question which I had was, I mean, actually the first 1,000 genomes, the first population sequencing, variation sequencing in humans and in other species was done also with low coverage sequencing. People forget. And we used imputation to basically improve the genotype calls. So, but that seems, I don't see that happening very often now. Do you use that? You know, what's the view about that in the PopGen community or in the?
Well, so I think we have like almost all the species we work on, we're basically starting from scratch and we have no reference panels or anything. We only have the data, the current data set that we're working on. So I think that limits our ability to impute a lot. But of course, there's several different approaches that people are starting to adopt. So some are doing sequencing a relatively small number of individuals at high coverage to generate a somewhat limited reference panel that they can then use to impute the rest of the data. There also is a software called Stitch that is developed specifically, and several other approaches too that can impute without reference panels, obviously. And we've been playing around with it a little bit on simulated data, and it works with, if you have large enough sample sizes, it can improve inference when you have high LD in your population. But as LD decreases, and also if you have pretty low SNP density, then the improvement that you can achieve becomes less and less. But I think for something like a recently bottlenecked population or something, there should be actually really good potential for being able to do reference-free imputation at this point, and it can actually improve inference quite a bit.
Okay. I think actually both Beagle and Impute will also impute without a reference panel.
Yeah, that's right. Mm-hmm.
So, but you're right, I think, about the LD. Okay, were there other questions? Let's see. Does anyone on the panel have a question? We can ask a question. I think otherwise, that's it. And it's the right time anyway. Thank you, everybody. And thank you, everyone who's watching. And we'll take a break now. And then I think after that, there's spatial chat and maybe some other prearranged meetings. Bye.
Thank you all for joining. Bye.